So nice to hear chatter before we start, so I hate to interrupt you. Just a reminder, I do have office hours on Friday by Zoom from 9 to 11 in the morning. Haven't had very little traffic on that Zoom meeting, so the link, it should be under the announcements. You might have to scroll back a little bit. Or usually I have the Zoom meeting on my iPad and I'm on my computer. So if you want to just send me an email, hey, can you send me the link? That's an easier way for you to hook up maybe as well. So take advantage of that if you have any questions or just want to chat. I do want to go back, revisit our last exam. Finally, we've got everybody taking it now, and just go over the top 5 questions that were missed. These were the only 5 questions that less than half of you missed. So this was the most missed question. I think like 37% of you got it correct, but there were maybe 3 or 4 questions that had to do with fermentation. So if that was something that you weren't as confident about, that might have been tough. But the main purpose of fermentation is to regenerate NAD. Okay, so basically the NAD that gets reduced to NADH from glycolysis, if those electrons from NADH don't have anywhere to dump off, then essentially glycolysis stops. Okay, and organisms that rely on fermentation rely on the ATP they get during glycolysis, because many fermentation pathways don't produce any additional ATP. All right, so again, if those NADHs don't have anywhere to drop off their electrons, then glycolysis ends up halting as well. Some confusion about this question: which of the following is not involved in the initiation of transcription? Obvious answer here is origin. The origin marks the beginning of replication. Okay, a number of you asked me, well, I don't know what consensus sequences are, so I just said that. I just put up the slide where we talked about consensus sequences. Remember the -10 region and the -35 region of the DNA of that promoter region. The more closely those align with some consensus sequence, the more likely RNA polymerase is to bind to the promoter, and the more likely then the gene is to get transcribed. We didn't spend much time on beta oxidation, but you had a slide that explained that as well. Remember, lipids are split into glycerol and fatty acids. And then when the fatty acids are catabolized during beta oxidation, basically 2 carbon pieces are cleaved off and bound to coenzyme A. And then that enters into the Krebs cycle, and we get lots of energy from that. So we talked about lots of energy from fatty acids because you get all these acetyl-CoAs from that. We skipped over most of our repair mechanisms, but we did talk about mismatch repair, and my arrow got kind of mixed up a little bit here somehow when I transferred it over. But during mismatch repair, the strand that's not methylated is repaired. So there's a little confusion about that. You had to maybe review or look at that diagram a little bit more closely, but the old strands are really highly methylated. And if an enzyme, repair enzyme detects a mismatch, the wrong base pair put in there, then it's going to fix the strand that is newer, okay? The one that's not been methylated yet. Eventually both strands get methylated, but the new one would not be methylated yet. Okay, and lastly, which of the following, I think this was the last one, which of the following is false about prokaryotic translation? Again, my arrow got moved a little bit here. Here, initiation begins with the recognition of the Pribnow box by RNA polymerase. That is true, but it is true for transcription and not translation. So I had to read the questions pretty carefully there. Okay, so I still have people coming by to look at their exams. I'll be in my office after class. If you want to take a look, feel free to do that. I thought I would throw up a question from last year's exam 3, because I often have a lot of questions. People say, well, what are you going to ask about these chemical disinfectants? Here's a question from last year's exam. So which of the following is an effective treatment to destroy endospores? So you had to kind of think about all the different methods we talked about. Remember, we organized our chemicals in terms of low, medium, and high. It was only those high antimicrobials that were considered sporicidal. Okay, and of course we talked about cold treatments, refrigeration and freezing don't kill microbes at all. They just keep them from growing. So it wouldn't be that. It's actually, the aldehydes is our high level that we talked about as sporicidal. Okay. So there's just an example. And when we finished last time, we were talking about therapeutic index. So I'm going to give you a minute to look at this question, and we'll just review that concept a little bit. Give you a second to read it through and pick an answer. Okay, so it might help to do the math a little bit. So drug A has a toxic dose of 1 and a therapeutic dose of 0.1. So that therapeutic index is going to be 10. Drug B, 0.5 toxic dose, 0.5 therapeutic dose. So drug B, the therapeutic index is going to be 1. Okay. The higher the therapeutic index, the safer it is. So the more acceptable therapeutic index is going to be drug A. Actually, 10 is really not a great therapeutic index, but just for the sake of definitions here, we'd like to see it much higher than that. But the principle there, the higher it is, then the safer it is. Okay. All right, so that's where we left off, talking about therapeutic index. Let's move on. We have a few more things to talk about in terms of kind of antibiotics in general and how they're used before we start into our specific drug discussion. All right, so there's really actually 4 different ways that the antibiotics are administered. This chart here leaves off thinking about topical administration. So some antibiotics we just put on our skin, okay? But other than that, there's three ways we can give antibiotics orally, meaning you swallow them either in liquid or pill form. They can be injected by syringe, or they can be administered in an IV. So there's kind of good things and bad things about each one of those methods. So given orally, obviously this is much easier for patients to take at home. The concentration in the tissues doesn't typically get as high when you take it orally. And you also have to think about kind of the safety of the drug. So when you're thinking about methods of administration, you have to keep in mind that therapeutic index. Okay, if the drug has to be dosed out just right, or else the patient starts getting toxic side effects, you don't want to give that drug orally, because then you're depending on the patient to take the drug at the right time, or not forget to take it and then take 2 or do something crazy like that. If the therapeutic index is low, almost always you're going to administer the drug by IV, because then you can dial it in basically to the concentration that exactly what's safe and will still kill the microbe. So you can get the concentration in the tissues up a lot higher with IV. by injection, you can get it higher than orally as well, but it tends to kind of dissipate pretty quickly after that. The other thing that you have to consider with administration methods is the susceptibility of the drug to stomach acid. As we'll see, some of our drugs basically are disrupted, their activity is disrupted by stomach acid. So then we can't give them orally. The only choice we have is by injection or by IV. Okay, all right. Of course, resistance is more and more of a concern. That resistance can either be intrinsic or acquired. So by intrinsic, I mean like a drug that targets mycolic acid is not going to have any effect on penicillin, or sorry, not on, say, gram-positive organisms. Because gram-positive organisms don't have mycolic acid in their cell walls. So a staphylococcus is going to be intrinsically resistant to a drug that targets mycolic acid. It doesn't have that structure. What worries us more is the acquired resistance that we see, a drug that for a while works on an organism and then after a while it doesn't work anymore because the organism has acquired some way to survive being in the presence of that drug. And we're going to talk a little bit later, probably won't get to it today, as some specific ways that organisms acquire that. Sometimes it's by mutation, sometimes by some kind of horizontal gene transfer. And so we'll talk about those specific methods soon. And then ideally, the drug that you want to use is inexpensive. Easy to mass produce and it doesn't cost very much. Otherwise, it's probably not going to be in widespread use if it's too expensive. All right, so we are going to start with a bunch of antibiotics that their mechanism of action is to inhibit cell wall synthesis. And there's a big group of organisms that are, keep getting it mixed up, a big group of drugs that do this, and we call them our beta-lactam antibiotics, or beta-lactams. Okay, so this is the group of drugs that includes penicillin. So they're kind of the most famous ones, probably. Why do we call them beta-lactams? Because they have this beta-lactam ring in their structure. Basically, if anything disrupts that beta-lactam ring, if any of these bonds are broken, then that antibiotic is not going to work. Okay, it's not going to be functional to kill bacteria. All right, so some other examples, carbapenems, cephalosporins, there's some semi-synthetic penicillins like methicillin. Okay, those are all considered beta-lactams. Okay. And I don't want you to be able to reproduce these structures. Like I couldn't do that, but you should recognize that beta-lactam ring. Okay, so here's a nice graphic from your book that explores in a little bit more detail how beta-lactams work. All right, they inhibit peptidoglycan formation by binding to particular enzymes. And we talked about these enzymes back when we were talking about cell structure and how cell walls grow. You might remember these enzymes are called transpeptidases. Okay, and what transpeptidases do, they actually cleave these amino acid cross bridges when the cell is adding new sugar subunits, NAM and NAG, into the peptidoglycan structure. And then it seals those amino acid cross bridges back up again. Okay, so that's what transpeptidase enzymes do. Certain enzymes called autolysins cleave the bonds between the sugar subunits, and then, but we were mostly focused on the transpeptidases. So the top here, you can kind of read this going across. So here's a blow up of the cell wall, how it is normally. And when it grows, right, you have to break some of these bonds here, add in new subunits, and then, you know, that's all orchestrated very carefully so that the cell wall structure stays intact. If the cell, though, is in the presence of some, say, penicillins, some beta-lactam, they just kind of put in these great big green blocks where there should be some amino acid cross bridges, and there's not, because the penicillin keeps those from forming. So you get a cell wall structure that looks a little bit wobbly or not as strong. And so the cell is especially susceptible to water rushing in, because the cell is often in a hypotonic medium. Okay, so you have the inrush of water due to turgor pressure, basically osmotic forces, and then the cell is really susceptible to bursting because the cell wall is not intact. Okay, so let's see, I didn't have room to write anything else there. So key points here are that beta-lactams work especially well, or let's say work best, on growing cells. Okay, if a cell is not growing, if its cell wall is not expanding, then peptidoglycan doesn't really have any effect in weakening those cell walls. So that's an important thing to keep in mind. And they also kill cells because they burst due to turgor pressure. Okay, the influx of water, because cells are often in some kind of solution where the solute concentration is lower than inside of the cell. So most often the net movement of water is going to be inside the cell. All right. So if we wanted to look at what was going on a molecular level, it is just mostly kind of out of interest to me as exactly what's going on. But so let me explain this. This D-ala structure, these two alanine amino acids, typically those peptide cross bridges in the structure of peptidoglycan typically are made up of two alanines side by side. And amino acids have a chirality. They're either kind of one direction or the other. And that's a concept that's kind of hard for me to wrap my mind around. But sometimes we think of it as a left-hand amino acid or a right-hand amino acid, even though it's not a perfect analogy. But there's actually, usually amino acids are in the L form, but for whatever reason, we find the D form of amino acids in bacterial cell walls, and that's kind of unusual. So anyway, that's a little bit of a side explanation. But so here's the structure again of two of the amino acids. If here's the structure of penicillin, all right? So when you superimpose those two structures, ooh, they're exactly the same, okay? And that's not an accident. Penicillin binds right there to those amino acid cross bridges, and that's the active site for the transpeptidase enzymes. Okay, so what do you call something that fits into the active sites of an enzyme? It's a competitive inhibitor. We actually don't call this a competitive inhibitor because it's permanent. Okay, so it's kind of like a competitive inhibitor, but again, because it's permanent and because the penicillin stays bound there, we don't typically refer to it as that. But okay, so penicillin blocks the active site so that the cell again can't grow normally. Okay. All right. So don't be afraid to jump in if you have questions or comments. Okay. Let's start with our penicillins that we would consider narrow spectrum. So the very first penicillin that was developed, it's called penicillin G. We had kind of different products of that original penicillin mold that they named, you know, by letters of the alphabet, A, B, C, D. Well, penicillin G had the best activity against bacteria, so that was the one that they mass produced. And the first penicillin was really narrow spectrum. It only, well, not really, but it only worked on gram positives. Okay, but that was a great advance, especially staph infections, streptococcus infections. It was great to have a drug again that worked on those kinds of infections. And because Chain and Flory did not patent the drug, it had extremely low cost right from the beginning. So it was available to many, many people right away. It penetrated into the tissues well, and there really were very few side effects. So this was really a miracle drug when it came out. Big disadvantage, really within a few years after it came out, we started to see the emergence of resistance. Okay, and so much now that penicillin is rarely used anymore because of resistant issues. What caused the resistance? Well, bacteria were able to produce an enzyme called beta-lactamase that actually made the antibiotic not effective anymore. So we're going to look a little bit more closely at the action of beta-lactamases in just a minute. So hold that thought. Another disadvantage is that because the drug was labile and acid, so in the stomach acid, it basically broke down, lost its structure. We have to inject it, all right? And even if it did pass through the stomach untouched, it's absorbed pretty poorly through the small intestine. So you would have to give a whole bunch of the drug. Well, even when we inject it, we have to give it a lot of it, again, because most of it just gets excreted with the urine rather than being absorbed. Okay. So, you know, my dad, you know, your grandparents were probably of the age that maybe don't remember when antibiotics come out, but, you know, my dad was a young child in the late 40s, and he remembers very clearly Doc Martin coming to his house and when he was sick and giving him an injection of penicillin in the rear end. You know, kids these days have it easy. Okay. Penicillin V then is still considered a natural product of that Penicillium chrysogenum. But if you grew that organism on a little different media, it gave a slightly different antibiotic that it secreted. Okay, so penicillin V is, again, the same organism grown on a different media. It's almost the same structure. If you compare the structures, the only difference here is penicillin V has an oxygen here instead of a carbon. Okay, so really similar, but that slight change in the structure made the the antibiotic more stable in acid, so that people were able to take it in pill or liquid form. Okay, absorption was still poor, so you had to take it, again, a lot of it. And then, I don't know where this arrow came from, so just ignore that. It just kind of popped up there. Then in the let's see, I think it was the early 60s is when methicillin was produced, and this is considered a semi-synthetic. So we took the penicillin and we were able to alter parts of it in the lab, basically made it a little bit bulkier side chain here so that it protected the active site a little bit better. And we were hoping that that would, organisms wouldn't become resistant to it as quickly. Well, in the early 60s, actually, we started seeing an organism that we have since named MRSA, or we named at that time MRSA, methicillin-resistant Staph aureus. So resistance did show up pretty quickly. Today, it seems weird that we call that organism that, because methicillin is a drug that's not in use anymore because, again, it's just a lot of things are so resistant to it. But that's where the name came from, methicillin-resistant Staph aureus. This was acid labile, so it had to be injected. Oh yeah, and then I talked about that down there. Okay. All right, so we're thinking about generations. So we had early generation penicillin, really narrow spectrum. As we came out with more and more variations of penicillin, we were able to get them a little bit more broad spectrum. Okay, amoxicillin came out in the early 1970s, and it's still in widespread use today. It's the one that's commonly taken whenever, you know, kids have an ear infection or strep throat. They put bubblegum flavoring in it, so kids don't mind taking their medication. I can remember in the 70s, when I took penicillin, it was like the worst tasting thing that you can imagine. You know, my mom would give me a cracker and say, here, this will make it taste better. It didn't do anything. It still tasted terrible. But again, a good thing about amoxicillin, you know, you can take it orally, less frequent doses than some of the earlier penicillins. Like I said, we're still using this today, but resistance, this common theme, is a problem. So today it's often marketed in the form, it's actually called augmentin. If you augment something, that means you add to it to make it better. So to amoxicillin, we've added clavulanic acid, which is a beta-lactamase inhibitor. Okay, so it's supposed to kind of shield that drug so that enzymes that bacteria produce can't, you know, break the drug down. Okay. All right, so let's talk a little bit more about beta-lactamase since I've mentioned it a few times. Oh, this didn't come through right. Okay, so sometimes beta-lactamase is referred to as penicillinase. It's the same enzyme. So here's penicillin. When this enzyme works on penicillin, what does it do? It actually cuts this bond right here in the beta-lactam ring and opens that up a little bit. And just that small structural change is enough so that penicillin doesn't fit into the active site of transpeptidase. Question?
Go back for a second.
Go back to this? Ah, okay. Yes, so clavulanic acid. Clavulanic acid it's actually an antibiotic itself, but we use it as a beta-lactamase inhibitor. So I don't know exactly the structural things of where that would bind to the antibiotic to keep this enzyme from working on it. But okay. All right, so again, beta-lactamase, sometimes it's easy to get confused about this. This is an enzyme that's produced by bacteria. okay? And it enables them to basically live in the presence of some beta-lactam. So how do they acquire the ability to make beta-lactamase? Usually that's some kind of horizontal gene transfer. They get the plasmid from another cell that has it. Okay. The other thing that I want to kind of stop and talk about is penicillin allergies. Most commonly, an allergy will manifest itself by rashes or hives. And remember, this is not really considered a side effect of the drug. It's an inappropriate immune reaction that our body has when we're given the drug. So again, sometimes it usually shows up first as just a little rash. About 10% of Americans are thought to be allergic to penicillin. So it's pretty widespread. I know for me, when I was 4 or 5, I got a little rash. Forever after, I'm not allowed to have penicillin or any beta-lactam antibiotic. And, I've often wondered, well, how bad? It was just a rash, it's no big thing. I'd much rather have the cheap antibiotic than having to take the expensive one. But you won't find a doctor that's willing to give it to you if you've had a rash, because oftentimes the second time or the third time you have the drug, your response can be even worse. Some people produce a special kind of antibody, an IgE antibody, that actually, it's the breakdown of penicillin when it's metabolized in our liver is what usually causes the reaction. And people that have that can actually go into anaphylactic shock. Basically, that means that the blood vessels get so leaky that their blood pressure goes way down, and it just causes basically all their organs to shut down. And about 300 people in the US every year die of an allergic reaction to penicillin. So it's something that doctors take pretty seriously. Some of you may know Dr. Baxter, have him for lab. He teaches microbiology as well. When he was a teenager, he had hives for 9 months after he had penicillin. So a little rash, a little hives, no big deal. But if you've ever had hives, or if you had something that looked like this, it's just can drive you crazy. Like, the itching is just awful. I wouldn't wish that on anybody. And to have it for 9 months, it's kind of even hard to think about that. So, okay. All right, so other beta-lactam drugs. Cephalosporins. Okay. Any drug that's a cephalosporin usually begins with that prefix ceph something. Cephalexin is probably the most common one. The trade name for cephalexin is Keflex. So it's kind of confusing. A lot of drugs have 2 different names, like the chemical thing that we call it, and then the company that makes it slaps its own name on there. So Keflex, again, you might have heard of that. So because it's a beta-lactam, it has the same mechanism of action, binds with transpeptidases. We have different generations of cephalosporins. So basically, it depends in terms of how they work and what they work on. It depends on the generation. So the earliest cephalosporin was very narrow spectrum and was not particularly toxic, had a pretty high therapeutic index, and we've made kind of different improvements on it. We're up to, I think, 5th generation cephalosporins. They have a broader spectrum, but tends to be a little bit higher toxicity. Okay, a little lower therapeutic index, so they have to be dosed just right. So the earlier ones can be I think this is supposed to be, the earlier ones can be taken orally. The later ones have to be injected. The later ones, oh, I chopped some of my slide off here. The later ones can actually cross the blood-brain barrier. So again, we have to dose that just right, but there are some infections that can do that, like meningitis. So we need to have an antibiotic that can follow the infection there. Okay, so let's see, 5th generation used to treat meningitis. Also MRSA. So methicillin-resistant Staph aureus is typically resistant to all the beta-lactam drugs. This is a beta-lactam, but again, the 5th generation, we've kind of monkeyed with it enough that it's different enough that often we see some activity against MRSA. Okay. And then I like to mention the carbapenems just because about 10 years ago, there was an outbreak in a hospital it was at an NIH health center, I think it was in Maryland, of a strain of bacteria that was found to be resistant to carbapenems. They called it KPC, Klebsiella pneumoniae carbapenem resistant. So Klebsiella is an organism that we work with in lab quite a bit. It's one that's not typically too much of a problem clinically, but you've got this strain that's resistant to carbapenems, and carbapenems are more likely used as kind of a last resort, kind of as an effort to keep this drug from becoming, you know, a lot of resistance happening to it. It is considered broad spectrum, but we only use it for gram-negatives because we have less options of drugs that work on gram-negative organisms. Okay, and we don't give it orally. Again, that's out of an effort to try to keep resistance from forming. So when we did have this outbreak and they found that it was due to this organism that was resistant to carbapenems, it was really a cause for alarm. And there's a really interesting documentary that PBS put out on Frontline a number of years ago that talked about antibiotic resistant superbugs. And this was one of the organisms that they featured. So if you ever get a chance to watch that, you know, it's Frontline. I think it was called superbugs. Okay. So that's it for the beta-lactams. A couple of other drugs. Oh man, totally cut off my bacitracin there. That's okay. I do have a couple other drugs that I want to mention that are not beta-lactams, but they still target the cell wall slightly different way. Vancomycin, you can see the huge bulky structure of vancomycin. It actually binds to that D-ALA, the 2 amino acid cross-bridge links, and just makes it so the transpeptidases can't get to it. Okay, so basically, again, the cell wall can't grow. A big physical obstruction. Narrow spectrum drug only works on gram-positive bacteria. This is one of the more, usually the first one that we go to treat MRSA. Also is used to treat Clostridium difficile, C. diff. And typically we have to give vancomycin by IV because it's not absorbed very well, but the exception of that is when we're treating C. diff. C. diff, remember, I think I've mentioned that maybe once or twice, is an organism that tends to overgrow, especially if a person has been on a heavy course of antibiotics, knocks out their other good bacteria. The C. diff overgrows and causes a really nasty diarrhea. So when we try to treat that with vancomycin, we have to swallow the vancomycin so it gets to the intestine where it needs to be. Okay, so it works better if that is swallowed. There are some toxicity issues with this drug, with the kidneys. So typically it's, again, not going to be given just for strep throat or something like that. You wouldn't use it until other things haven't worked. And so we even have some bacteria that have found to be resistant to vancomycin. This is another, if you have what's called a VRE infection, vancomycin resistant enterococci, that's a medical emergency because we don't really have anything left if that's not working. All right, bacitracin, let's see, hopefully you have this on your slide here. Sorry, it got cut off of mine. How it works, so those new NAM and NAG subunits for peptidoglycan are made inside of the cell and then transported out to the cell wall. Bacitracin basically blocks the movement of those new subunits. So again, it's keeping the cell wall from expanding. Let's see, what else did I have to say about this? Not too much else. It's pretty narrow spectrum. I guess the big thing to remember about bacitracin is because it's pretty toxic to the kidneys, it's only given topically. Okay, so it's used only for skin infections. But you have probably used bacitracin quite a bit in your life because it is in triple antibiotic cream. Okay, it's one of the antibiotics in triple antibiotic cream. So again, cell wall inhibitor. It's the most common place that you're going to see that. All right, and lastly, of the cell wall inhibitors, isoniazid. I think that one's just kind of fun to say. Isoniazid, it's the one that I mentioned before that prevents the synthesis of mycolic acid. So gram-positives and gram-negatives don't have mycolic acid in their cell wall, but this is going to be active against our acid-fast organisms, the mycobacteria. All right, so we're going to be using isoniazid for tuberculosis and leprosy, are two of the more common infections caused by mycobacteria. Okay, and this actually is a synthetic drug, so it's not biosynthesized like a lot of our other drugs. Okay, all right, so I left a blank page here because I want to start you on a concept map to try to help you keep some of these drugs straight, because we are doing about 15 of them. So if you've got a blank piece of paper somewhere, a full page is better because we're going to add to this as we go, or turn page over or whatever. So if we take, let's see, I might have to. Okay, so if we start with our cell wall inhibitors, and our beta-lactams are a big group of cell wall inhibitors. Okay, so then we could list these. Let's see if I gave myself room to write them or not. Penicillin V, penicillin G was the first one, and then penicillin V. We talked about methicillin, amoxicillin, carbapenems, cephalosporins. Okay, so those were our beta-lactams that we talked about. Of course, there's lots more. These were just the ones that we chose. Okay, and then there were other cell wall inhibitors like vancomycin and bacitracin and isoniazid. Okay, oh, looks a little bit sloppy. But we're going to add to this our next mechanism that we're going to cover is protein synthesis inhibitors. Okay. So I'll stop there and we'll add on to that as we go through the rest of our drugs. Okay, and you could add on to this, you know, like any weird side effects we talked about, or if you want to get a little bit more specific with the mechanism, you know, like isoniazids, you know, targets mycolic acid, you might have a little bit more room to write things in. But if you can, you know, for the exam that's coming up in a couple weeks, if you can kind of have this pictured in your mind, or maybe practice spitting this out, if you can remember the mechanism of action for all of these drugs, then that's going to be a really good first step to be successful answering questions on the exam. Okay. All right. So we will come back to this, and I can post it later, like totally filled out as well. Okay. So let's keep moving a little bit here. That's all right. All right, so our second biggest class of drugs that we're going to talk about here are the protein synthesis inhibitors. So most of these are all targeting bacterial ribosomes. Most of them are actually targeting the small subunit, that 30S subunit. And most of them are pretty broad spectrum. So they're killing both gram positives and gram negatives. And remember, we talked about the different organisms briefly that produce antibiotics. Streptomyces is that genera of bacteria. It's a soil organism that kind of acts like a fungus, but still prokaryotic. That's a huge producer of drugs in this class. So a lot of our protein synthesis inhibitors come from some species of Streptomyces. Okay, so one big class of protein synthesis inhibitors are the aminoglycosides. They bind to the 30S subunit of bacterial ribosome, which, remember, bacterial ribosome, different structure than our eukaryotic ribosomes. They're in general pretty broad spectrum. I already said that. They don't absorb well, and they're labile to acid, so we have to inject them. And another drawback is that they have pretty high toxicity, and that's manifested mostly by toxicity to the kidneys as well as to the ears. So I found this little kind of a mnemonic of a kidney boxing with an ear. You've got the names of some of the aminoglycosides like gentamicin, tobramycin on the gloves there. Okay, ototoxicity and nephrotoxicity. Again, some side effects here to the kidneys and ears. And these drugs typically end in myosin. All right, so kanamycin, gentamicin, streptomycin, we're going to talk about this one in detail. Neomycin. Gentamicin is spelled differently because it's a natural product of a different species of organism, in case you're wondered. Neomycin, we're not going to talk about this one anymore, but this is the second drug in triple antibiotic cream. Okay, so bacitracin, cell wall inhibitor, neomycin, protein synthesis inhibitor. The third is a third mechanism that we'll get to a little bit later. All right, so let's talk about streptomycin a little bit. This is an example of an aminoglycoside, and I like to put structures up here, not because I want you to memorize them, but oftentimes, you know, the name kind of reflects what the structure is, and it kind of makes a little bit more sense. So aminoglycosides, they're amino-modified glycosidic sugars. So when you look at the structure here, this is another way to draw sugars. So you can see the sugars here. Amino-modified, here's lots of amino groups here. So that's where the name aminoglycosides came from. And streptomycin is probably the most famous of the aminoglycosides. It was discovered from a soil sample in mid-1940s. I think we came out with this in like 1946 or 47. The guy's name was Selman Wakeman. He went on to discover about 20 different antibiotics in his lab. And we know now, though, that he took all the credit, and it was a PhD student in his lab that actually discovered it, and he took all the credit for it. So interesting story there. But streptomycin works because it actually binds to the small subunit of the ribosome, changes its shape just enough so that the incoming tRNAs don't recognize that messenger RNA accurately, and it brings in the wrong amino acid. Okay, so again, messing with protein synthesis. Broad spectrum drug. The toxicity to the ears, that seems like a weird side effect. If you are in anatomy, this will make more sense to you, but it actually binds to the vestibular portion of cranial nerve number 8. So again, you all that are in anatomy, that will maybe make a little bit more sense. If you're not in anatomy, don't worry about that detail. What kind of side effects does that cause? It might be something as simple as a little ringing in the ears, tinnitus, that goes away. It might cause, what do you call that? The vertigo, where a person basically is nauseous all the time because of, you know, some inner ear issue. That typically would go away as well, even though that's really hard to endure. It can actually cause hearing loss, even all the way mild, all the way up to deafness, which can be permanent. So for that reason, we don't give streptomycin unless nothing else is working. It was really a miracle drug when it came out, though, because it was the first drug that had any activity against tuberculosis. All right, so up to that point, you know, we've had tuberculosis for thousands of years, and suddenly in 1946, we had a drug that would cure people of tuberculosis. So it was really a miracle drug. Again, had to be really careful about trying to dose that out right so that toxic effects, you didn't have too much of that. But when you're trying to save somebody's life, then some of that is okay. So it was also used in some other really severe infections like bubonic plague, tularemia. That's an infection that's, I can't remember if we talked about that one or not, but that's passed by the fleas on rabbits, and that can be spread from person to person in a pneumonic form. So that can be a very serious disease as well. Okay. Other protein synthesis inhibitors, tetracyclines, that's a whole nother class. So we're just going to talk about that kind of class in general. The name tetracycline, again, here makes sense. 4 rings, tetracycline, the structure there. So these are not aminoglycosides. This is a different kind of class of protein synthesis inhibitor. They also target the small subunit in a little different way. I think my next slide shows that in a little, I again, different way. But they are also broad spectrum, semi-synthetic, and they're really cheap. So for that reason, they have been really quite overused in agriculture. It was realized sometime 1960s or 70s that when you give livestock feed with antibiotics in it, it makes them grow faster. Right, so pretty quickly, all livestock feed had antibiotics in it. Particularly in chickens, we've found that salmonella, which is kind of part of the normal microbiota now of chickens, all the salmonella is resistant to tetracycline. So we think that that's, you know, really been overused. Problem is, profit margins are pretty small in agriculture, so anything that gave farmers a little boost, basically they had to do it to compete with other farmers. So it's kind of like this huge problem that's a tough one to solve. California now has banned the use of antibiotics. I know they did that a few years back, and maybe other states have followed. I'm not sure. I'd have to check about that. What do we use tetracyclines for? There's typically adults only because of the side effects that you can't miss this picture here. So tetracycline affects developing bones and teeth. Got cut off of my slide there, sorry. So we don't give tetracycline to children just because obviously we don't want their teeth to end up gray like this, or having their bones not be as strong, or maybe even misformed. So, but adults can take tetracycline pretty safely. It's common in acne medications, a few other specific infections. Besides affecting developing bones and teeth, it also has a phototoxicity. So if you happen to be on an acne medication with tetracycline in it, your doctor's going to tell you, whenever you go outside in the sun, make sure you are slathered with sunscreen, because you will get sunburn much more easily if you are on a medication with tetracycline in it. And it also, interestingly enough, with all drugs, you know, they have a use by date. And, you know, I tend to think, well, you know, I can go past that a little bit. Usually they say use by a certain date because it kind of will degrade after that and not be as effective. But actually tetracycline, when it gets old, it actually changes into a form that's even more toxic. So in general, you shouldn't take drugs past their use by date, especially a tetracycline. Okay, so just a little graphic of the mechanism of action. It actually binds right to that docking site of messenger RNA. So it's kind of a physical block for those tRNAs that are bringing in amino acids. It's more of a static drug. So basically just keeps the organism from growing, from making new proteins. Once the concentration goes down enough, then the organism could start making proteins again. But again, hopefully, if you keep the organism from growing and reproducing, then our immune system gives it a chance to move in and totally get rid of the bacteria. All right, one more protein synthesis inhibitor, a big, not a big, anyway, a class of drugs that includes at least several, are the macrolides. So this apparently is a macrolide ring. That's a little bit harder to kind of discern that. Probably the most common macrolide that you would have heard of is azithromycin. It's also called a Z-Pak. So it's actually in 2010, this was the most commonly prescribed drug for kind of outpatients, outpatients that were ill. So it's really commonly prescribed drug, especially for people that are allergic and can't take penicillin. Azithromycin is probably usually the next choice. It targets the large subunits of the ribosomes. Okay, so this graphic from your book, also mentions some drugs that we're not worrying about. There may be a few homework questions that mention drugs that we're not going to talk about in class. You don't have to worry about those for the exam, but again, sometimes a homework question has several parts, and I couldn't get rid of the part that you didn't need to know, so you might have to look a couple things up for homework questions in the book. Yeah, it's just kind of binds to the messenger RNA there, and can't, move through the ribosome to be accurately translating proteins. All right, so common infections like ear infections, strep throat. It's one of the drugs that we use to treat gonorrhea. That is constantly changing because gonorrhea is so resistant, and over the years, so we have to keep changing, but we use it in combination. It's one of the drugs given to treat gonorrhea. Some gram positives and some gram negatives are susceptible to it. Really nice thing about azithromycin is that it basically gets absorbed well into tissues, and it stays active for a long time. So you only have to take it once a day, and usually only have to take it for 5 days. So that's kind of a nice thing. There are some allergies to azithromycin, some GI issues like a little nausea, a little bit of cramping, but not as much as erythromycin, which is another macrolide that's what I used to have to take when I was a kid, and I can just remember just not feeling like I could eat anything. Thankful that I got rid of the strep throat, but kind of still felt lousy for a while just because of the antibiotic. I also mentioned azithromycin has been in the news in the last 6 months or so because early on, it was one of the drugs that they were using or trying to use or seeing if it would work against coronavirus. And that really stumped me, like, this virus is not going to work on viruses. It's an antibiotic. But as I kind of dug into that, it turns out that azithromycin actually has some activity against some viruses. And I don't understand the exact mechanism of that other than they say that it keeps some viruses from entering host cells. So that's kind of interesting. And it also can kind of boost our immune system. They think that azithromycin increases the production of interferons, which are cytokines that help, it's kind of our non-specific immunity against viral infections, and that it may tamp down inflammation as well. So we know with coronavirus, sometimes we just get a huge amount of inflammation. That's kind of our body's response to the virus, and that needs to be tamped down. It's just kind of an inappropriate response. So I don't think that they decided that erythromycin was really kind of worth it, but I know that they were trying that for a while. So, okay. All right, so moving on, let's talk about a RNA inhibitor first. Okay, rifampin. That probably is one that you may not have heard of before, but it does block RNA polymerase. I think that it binds to that enzyme during elongation of transcription. And again, just halts the flow of information there from DNA to RNA to proteins. This one has a really interesting side effect. Other than some kind of nausea that it can cause, it turns any bodily fluid orange. So urine, sweat, or tears. So here's an example of a catheter bag, somebody in the hospital that's on rifampin. Kind of shocking side effect there. Pretty harmless, but just kind of startling to see somebody's urine that orange. Why does it do that? Because the drug itself is kind of a dark orange color, and I guess it just, I don't know, keeps that. Uses. This is one of the only drugs that's absorbed well by Mycobacterium. So it's used for tuberculosis and leprosy, usually in combination with isoniazid, the one that targets mycolic acid. Okay, we'll talk about tuberculosis a little bit later in the semester, but we usually hit that infection with a combination of about 4 different drugs because we have so much problem with the Mycobacterium becoming resistant to drugs. Okay. And a drug that, oh, and by the way, I wanted to mention, remember RNA polymerases are RNA polymerases. We have a different RNA polymerase. I think I mentioned this in passing a few weeks ago that makes tRNA, a different one that makes messenger RNA, a different one that makes ribosomal RNA. Bacteria have, their RNA polymerase is different enough that the drug can, again, target the bacterial enzyme and not ours. Okay, so drugs that target DNA replication, quinolones and fluoroquinolones. So they target DNA gyrase. So if you remember, here's a replication fork. Remember, DNA gyrase works upstream here of the replication fork on the supercoiling issue. So as the replication fork opens, that DNA upstream gets all twisted up. The DNA gyrase comes in, clips it, unwinds it, and seals it up again. So if these drugs are bound to DNA gyrase, then that enzyme can't function and the DNA can actually break upstream of the replication fork. So that's how it harms bacteria. Kind of the quinolones were the early drugs that targeted DNA gyrase. And remember, that's different enough from bacteria, from enzymes that we have that kind of do the same thing. Quinolones were kind of the early version. Nalidixic acid was the first one. And that might ring a bell if you're in lab because we've used nalidixic acid. We put it in some blood agar. It's one of the drugs that we use in our selective media so that only gram positives can grow on our blood agar, colistin and nalidixic acid. So this was very narrow spectrum. This only worked on gram negatives. Fluoroquinolones then became a little bit more broad spectrum. Not much use for quinolones anymore just because of, I think, some toxicity issues. We found, though, that when we added a fluorine right here, we call those fluoroquinolones, and that just increased the efficacy a little bit of the quinolones, made them a little bit more broad spectrum as well, improved penetration into cells. So probably the most easily recognizable fluoroquinoline is ciprofloxacin, okay? And the fluoroquinolones always end in this oxacin. So when you see that suffix, it makes you think fluoroquinolone. Incidentally, another quinolone is not nalidixic acid, novabiacin. So that's another antibiotic that we've recently used in lab when we're trying to identify the staphylococcus. So, okay, so ciprofloxacin, we usually just call that Cipro. You may have heard that. That's the drug that's typically given to somebody if they're exposed to anthrax. So it's been a while ago now, 20 years ago since those bioterrorist attacks, but lots of people in the Capitol building and the Postal Service got to take Cipro for a week or two. Okay, let's see, readily absorbed, stable in acids, so it can be given orally. We don't use fluoroquinolines though for your garden variety infections. So you're not going to give this to kids that have strep throat or an ear infection. They're kind of used when other things don't work, maybe for respiratory infections or sexually transmitted disease, urinary tract infections. The reason is they have some kind of serious side effects in kids, or infants have gotten seizures. There's some kind of neurological side effects, headaches, dizziness in kids. So they don't give these to kids. And in adults, the kind of interesting side effects that often, not often, but when it shows up, it's a little bit more serious, is tendon disruption. So the Achilles tendon can actually be, I don't know if it will break, but you can have some injury to that Achilles tendon. It's not really clear why that happens, whether the drug limits the blood flow to the tendons or whether it impacts collagen formation. It's not really clear, but you know, usually if something else would work, then we would use something else first. Oh, and I found, just kind of surfing the web, I found this mnemonic that might help you remember how quinolones work. So the queen, the quinolones, stops the gyrating dancers, DNA gyrase. All right, so I don't know if that will help or not. I thought I'd throw that out. All right, 2 more drugs to go. Question? Yeah. Yes, so rifampin, we still call it an antibiotic, even though, did I say that it was synthetic? Okay, so the question is, I didn't specify whether it was broad or narrow spectrum. I guess I just dropped that detail off of this because I didn't have it memorized and it wasn't something that I thought you needed to concentrate on. So yeah, I don't know. Does that answer your question? Yeah. I believe it is. I didn't write that down here, but right. So even if a drug targets bacteria, we still call it an antibiotic, even if it is synthetic. Yeah, so we use that term a little bit loosely. Okay. All right, let's finish up with the polymyxins. These are drugs that target cell membranes. Okay, so looking at its structure here, they've got this tail that will insert right into cell membranes and disrupt them. So kind of a surfactant action, almost like soaps or detergents. They are more active against gram-negatives. So that outer membrane of gram-negatives, they're going to be really effective. If we think about it, our membranes are very similar to bacteria membranes, so they really have limited use because of the toxicity issues here. So we only use them topically. Okay, they're toxic to the kidney. Okay, colistin is an example here. So that's another one that you'll recognize if you're in lab, the colistin and the nalidixic acid that again inhibits gram-negatives from growing. All right, polymyxin B though, that's the third antibiotic in that triple antibiotic ointment. So one that targets the cell wall, one that targets protein synthesis, and targets bacterial membranes. So it's a good treatment if you ever get a scrape or whatever. Not much is going to survive that. All right, so we will finish up with sulfa drugs next time, talk a little bit about resistance, and then start into chapter 14, the process of infection. All right, have a good day.